So first, let me thank the organizer for first the terrific idea of organizing this meeting, and the program seemed absolutely fabulous, and I'm really thrilled to be here today and to be able to tell you a little bit about the neurobiology of gendered behavior, or sex-specific behavior. Now, my lab is interested in social behavior. What are the brain circuits that drive social behavior? And why social behavior? Because I think this is something that is very important for animals in general, for humans specifically. We like to be in groups, and both humans and animals in general usually do very poorly when they are kept isolated without their conspecific. So there is something very special about the brain areas and the circuit that process social information. Now, what is quite interesting is throughout the animal kingdom, social behavior is usually extremely sexually dimorphic, which is that males and females not only are very often morphologically different, but they also receive and respond to social cues in very different manners. And so that raises a very interesting question on how different are the male and the female brain. What generates these different responses to social cues and these different behaviors? So basically, animals are quite different from each other within any animal species, but the biggest differences really are differences between males and females. And so where does this originate from in the brain? Well, there are different views of this, and one obviously is that the male and the female brain are dramatically different. They are just built differently, and they respond to social cues differently, and that's the way it is. And this obviously cartoon, and many of you might have seen other forms of this cartoon provide a very exaggerated picture of potential differences between the male and the female brain. But it's important to look at it and think about those representations, because this has actually been the intellectual basis of most of the neurobiology of sex-specific behavior. And what I mean by this is that for now decades, neuroscientists have looked at anatomical differences in the brain between the male and the female brain. And there are differences. So for example, these are differences in the number of synaptic boutons in one particular area of the brain between males and females, so that's in the medial amygdala. There are also differences such as the number of neurons in specific brain area. This is the medial preoptic area that has one nucleus that is larger in males than in females. So there are anatomical differences. But here's the problem. Those differences are actually quite minor. Which is they seem to be too subtle, or at least amazingly subtle, to have explanatory power on the differences on behavior. And the second, more profound problem is that these differences are not seen in all species, although the differences in behavior do exist. So for example, this particular nucleus here that is larger in male than in female in many species of rodents is actually not sexually dimorphic in some animal species, although again, the differences in behavior do exist. And so more recently, analysis that have been done in human, both anatomical as well as functional studies, seem to show that categorical differences in brain areas between males and females cannot be seen, and that instead the human brain is a mosaic. And I'll come back a little bit to that study a bit later. So I think the fundamental problem with all these anatomical studies as explaining differences in behaviors is that we neuroscientists really don't know how to infer the structure, the function of a particular brain area according to its structure. In other words, what has been done so far is to correlate differences in structures with differences in function, but we really don't know the causative effect of those structural differences. And I think that some of the new neurobiology of gender that I will present today are really taking a very different approach and, interestingly, coming to extremely different conclusions. And this is what I would like to present today. Now, my lab works with mice, and I've been so far extremely reluctant to infer any broader conclusion beside mice on the type of results we have obtained. But as it turned out, there are quite a number of recent studies, both in psychology and neuroscience in other species, that really seem to bring a new picture of how the brain controls sex-specific behavior. And so I would like to try to present some of the work of other colleagues as well. Obviously, these could be seen as very questionable. Again, I'm a mouse geneticist, and I just think that some of the other studies are really interesting to look in this context. So let me start by saying that, again, what we are interested in are the neurobiology of instinctive social behaviors. And I think that what is really important to recognize is that there are different steps in the control of social behavior. One is the detection of social cues, which is how does an animal detect another animal and understand whether this animal is a male or a female or an infant or an adult. And this detection of social cues is very different from one species to the other. So birds rely a lot on social cues, rodents on chemical cues or pheromones, and humans rely a lot on visual and auditory cues. So this is really very distinct from one species to the other. But what is very clear, though, is that the circuit underlying those the processing of these cues and the modulation of these cues according to the physiological state, and then the areas that really are involved in triggering specific behaviors, those are thought to be very conserved across animal species. So the study that we're doing in mice that rely mainly on chemical cues, we think can have a lot of significance for the understanding of brain circuit, and these brain circuits might be conserved across animals. So what I would like to tell you today is some of our study that addresses the issue of how do animals recognize the gender of their conspecific, as well as what are the brain areas that control gender identity, and how is the control of gender-specific behavior achieved. So in most mammals, but not in humans, as I will come back in a few minutes, rely on chemical cues to detect their conspecific. And what you see here is called the Flehmen response, which is animal trying to capture social scent. And if they have this very specific facial expression, it's because they are trying to have social scent reach a particular set of olfactory neurons in a structure called the voronasal organ. These voronasal organs send input to a dedicated pathway in the brain that reaches the hypothalamus, which are the areas involved in the control of social behavior, such as mating, reproduction, and aggressive behavior. And these differ from another set of olfactory neurons called the main olfactory epithelium, in which neurons are sensitive to volatile cues that are just volatile odorants in general, and that are then sent largely to the olfactory cortex. So the voronasal organ has traditionally been seen as the core, the essential structure for detecting social cues and triggering social behaviors such as mating and aggression. And quite a while ago in my lab, we used genetic tools to really address the specific roles of these two olfactory systems. And for these, we used the fact that these two sets of olfactory neurons use very different signal transduction pathway. In brief, neurons from the voronasal organ use this ion channel called TRPC2, and neurons from the olfactory system use this other channel, which is an olfactory cyclic nucleotide gated channel. So the idea is that by deleting genetically one channel or the other, we can impair the function of the olfactory system or the voronasal system, and in turn look at the impact on social behavior. And to our big surprise, when we deleted genetically the TRPC2 ion channel in males, we realized that these males were perfectly able to mate normally. So in other words, the voronasal system does not seem to be essential for mating behavior. However, when we ablated the olfactory cyclic nucleotide gated channel, these animals were totally unable to mate. In other words, the main olfactory system in the mouse seems to be essential for mating behavior. However, when we looked at the other social behavior in this TRPC2 mutant, we observed something very interesting, which is that male mutants, instead of attacking other males, as a wild-type male would do, would attempt to mate with these males. And so they actually seem to be able to mate both with males and females, and when both males and females were put in a cage, this mutant male will attempt to mate with each sex with equal frequency. And so these suggested that the two olfactory systems are essential for the control of normal mating behavior. And what the voronasal system does is to be essential for sex discrimination. Now, as we were at the time observing these results, I start to worry that people would infer this result on all sort of human behavior. And so I tried to look at how other animals were able to discriminate the sex of conspecific. And I came to some really interesting observations. So these birds, for example, the American flicker, rely purely on visual cues to detect a male from a female. So this is the face of a female, this is the face of a male, and the male has this little black feather here that look a little bit like a mustache. And what happened is that if you paint this mustache on the face of a female, then the other male, instead of attempting to mate with that female, will now attack it because they think it's a male. And similarly, if you paint over this mustache here, now this other male will attempt to mate with this male without a mustache because they think it's a female. So in other words, I have a mustache, I'm a male. I don't have a mustache, I'm a female. Similarly, the parakeet here has one distinctive sign of its gender, which is the presence of a blue dot on the top of a beak. And here again, if you paint a blue dot on a female, it will be assumed to be a male. If you remove the blue dot from a male, it is assumed to be a female. And so Timbergen, a very famous ethologist, called these the badges of masculinity. You know, I have a blue dot, I'm a male, or I have a mustache, I'm a male. And so what we think we found is that the pheromones in mice are the equivalent, the olfactory equivalent of the badges of masculinity. Now, what about humans, obviously? Well, in human, this ion channel, the TRPC2 ion channel, is a pseudogene. And what you see with this little yellow triangle here are deleterious mutations that emerge at the root of the emergence of the old world monkeys, which interestingly correspond to the duplication of the red and the green opsin gene. So in other words, as the visual world expanded in these higher primates, then the sense of olfaction and the sense of VNO detection basically got reduced, and even disappeared. So the conclusion from this is that higher primate highly rely on vision rather than olfaction in detecting social cues. So in other words, is anything I've told you so far of any relevance to human? Well, I think that what I found quite interesting, that point to my attention, is that visual, olfactory cues are not essential for sex discrimination, but visual cues and auditory cues are actually extremely efficient for sexual arousal. And this is actually the entire power of pornography is based on this, which is that just providing the right visual and auditory cues are just sufficient to trigger neuronal circuit involved in mating behavior, which I found quite interesting. So now let's look now at another issue, that of gender identity, which is what and how, what brain areas and how are brain areas involved in controlling whether a female will behave like a female or male like a male. And interestingly, we actually didn't particularly look for that set of questions, and we just stumbled across a number of really interesting observations when we, instead of looking at male mutants, now looked at female mutants. And I have to tell you that as a young head of a lab many years ago, I had absolutely determined that we would do studies of social behavior both in males and in females. And we started, and then we completely stopped the work on females. We just were working only in males, and it became extremely embarrassing at meetings that I would describe the phenotype of male mutant, and then there would always be a woman in the audience raising her hand and say, so what about the phenotype of females? And it took us years to come to those studies, in part because there's very little literature on the behavior of females. And these come from a long time ago, and I found this quote extremely interesting. Compared with Darwin's exquisitely detailed observation of barnacle, coral reef, and orchids, even the emotional development of his own children, this consummate naturalist observation of women and other female primates were curiously cursory. So basically, there's very little interest in the behavior of females in general. The literature has very few studies of female behavior. And so if you want to undertake some of these studies, there's just very little references to look at. And as you may know, this is a much broader problem in biology, where most of the models that are being used to look at biological problems are done in male models. So I finally found a postdoc who was courageous enough to look at the behavior of females, and I think that it paid off quite well, which is that she found something extraordinarily interesting. But before I go into this, let me remind you, for those who don't know, many of you will know, how biological sex is established. And so, as you know, male and female have a difference in the sex chromosome. Male have a Y chromosome, and on the Y is an interesting gene called SRY, or the sex-determining region, which will be essential in determining the undifferentiated gonad and having them differentiate in males into the testes, and the females that do not have this SRY gene will have their gonads differentiating into ovaries. In turn, the ovaries and the testes are secreting different sex-specific hormones, and these sex-specific hormones are thought to act not only on the body, to feminize or masculinize the body, but also on the brain to establish male and female specific behavior. Now, interestingly, the level of hormones in males and females, the differences start very early. In human, there is a peak of testosterone happening in utero, and that level of testosterone in the fetus is actually as high of that of a sexually mature adult. So extremely high level of testosterone. This is transient, it disappears and then reappears right around birth. And then the level of testosterone stays very low until puberty, when there's an emergence of adult sexual traits. So the idea is that this early phase here organizes the male brain, but the absence of testosterone in the subsequent period has this circuit being non-functional or non-active until the emergence of puberty. And females do not have any secretion, notable secretion of steroids until sexual puberty, when now they start to cyclate the secretion of estrogen. And so the idea is that this is the basis of the emergence of the male and the female brain, which is the young male brain is influenced by this early secretion of testosterone and will organize male-specific circuit. And the female that does not, sorry, the male is here. So male has this very specific sex-specific circuit that drive male-specific behavior. And the female that do not have this secretion of testosterone now generate a different set of circuits. So in other words, the brain has a profoundly sexually dimorphic neuronal circuit. And now let me show you the data from Kali Kimchi, the postdoc who looked at the female behavior and questioned whether these hypotheses are true or not. I should say they are not even hypotheses. They are just the model of male versus female brain differentiation. So what Tali did is to look at the behavior of a female mutant for the TRPC2 ion channel. So again, this is an animal that is unable to detect pheromones, and the female is here in gray, and in black is a wild-type male. What she attempted to do here is something very simple, just look at sexual receptivity of this mutant female. And to her surprise, here's what she saw. Uh-oh, we're having a little problem here. Okay. So as you can see, this female absolutely does not behave like a female. It behaves exactly like a male. And you know, believe me, when I first saw this result, I was astonished because I couldn't comprehend what I was looking at. I went and checked that female. This is a female. And the behavior is extraordinarily reproducible. I must say that not only the attempt to mount this animal, this other conspecific like a male, but they have other male-like behavior. For example, they emit ultrasound, which normally only male emit. So they really have their entire repertoire of social behavior are now male-like. They also have reduction of female-specific behaviors such as maternal behavior. Importantly, we remove the vomeronasal organ in adult female to make sure what we're observing were not developmental defect, and we found exactly the same phenotype. And also, very importantly, these animal mutant or surgically ablated animal have a very normal level of steroid hormones. So what that says is something very different from what the model we've seen before, which is that this model in which only male have the neuronal circuit that drive male-like behavior, this model could not explain the phenotype that we have observed. And so what we propose instead is that, in fact, the male and the female brain, and the female brain to start with, have components of both male and female behavior, and that these circuits are driven or controlled by the vomeronasal system in the mouse. So there's a very dedicated area of system in the mouse that makes sure that female behave like female and repress neuronal circuit that drive male behavior. But the essential message here is that the male and female-like circuit are both present in the female brain. And so by parallelism, obviously, we propose that the male brain also has both male and female specific behavior circuit. And so is that true? Well, in subsequent work, we actually were able to demonstrate that this is the case. Let me show you those data. And the female specific behavior that we attempt to observe in men is parental behavior. Now, in mammals, moms are taking the primary responsibility for taking care, nurturing infants, and it makes a lot of sense evolutionary, since mammals have the female nurture the embryo, the fetus in utero for a long time, and once they are born, they are lactating, and therefore a female really have a primary role. Males in mammals have very different roles according to the species, For most of them, most part, they are just ignoring infants, and they are also often infanticidal, which is that they will kill pups or infants when they are encountering them. And this is particularly true for virgin males as well as foreign males, so males that belong to other groups. And when they are confronted with infants, they will kill them in order to have access to the female and sire their own infant. So we, in mice, male mice are robustly infanticidal. If you put a pup, they will immediately attack and kill them. And so we wondered whether this male-specific behavior would disappear in mutant males for the TRPC2 channel, and that instead they could display parental behavior. And this is indeed exactly what we observed. This is a TRPC2 male. It's a virgin male, and these virgin males are normally infanticidal. But in this case, we have a mutant animal for the TRPC2 ion channel. And what you see here are a bunch of pups. And so this mutant male already does something unusual, which is it's building a nest. Then approaching the pups, retrieving a pup, bringing it to the nest, and the video will stop here, but in the next few minutes, this male will basically go back and forth, retrieve all the pups, bring them to the nest, groom them, and crouch around them exactly like mom would do. So the only two differences is that the behavior of males are a little bit slower than that of a female, and also don't lactate, which obviously they can't do. So, so this is interesting. It suggests again that the males have absolutely the ability in their brain to be parental. And so the question is, well, why do they have this female-specific circuit in the brain and they are not using it? Well, as it turns out, they do. They do in particular circumstances when they become dad. So as it turns out, males in mice are infanticidal, but when they mate with a female, exactly 3 weeks after mating with a female, they are now paternal. And let me show that. This is a certified infanticidal male. We presented with pups and it attacked them. We had that male mate with a female, and now we are testing the behavior of these males 3 weeks later. And here's what's happening. Take one pup. So you can see, very efficient. Can also build a nest, as you can see. Last one. Now here's what's happening. Good dad, making sure nobody's left behind. Yeah. Coming back, grooming the pups, and then taking care of them. So that's just, transformation is absolutely amazing. And if you think about from an evolutionary perspective or a behavioral perspective, it's actually quite striking that if you look at insects, fish, amphibians, reptiles, birds, and mammals, in all these species, there are some of the species in which mom is the primary caregiver, some species in which both male and female are the caregiver, and some in which only the females are caregiver. So for example, and this, among some of these groups, for example, in amphibians, these poison frogs are genetically very similar to each other, but one is entirely maternal, one is entirely paternal. You can actually see the tadpole on the back here of this male, and some are biparental. So from a genetic standpoint, these species are very similar. They probably have exactly the same brain, or very similar, but however, their behavior is dramatically different in terms of sex-specific behavior, such as parental behavior. So these really led us to think that there must be a set of neurons in the brain of both males and females that drive parental behavior, and that it's not the presence of these neurons that is different between males and females. It's more the way the function of these neurons is modulated. And so we decided to try to look at neurons activated during parental behavior, both in males and in females. For this, we use a molecular readout, which is that we have an animal interacting with pups, and we know that when neurons fire, so when they are active, they express a particular gene called CFOS. And so we then looked at both males and females interacting with pups, and then look at the expression of CFOS in the brain of parental animal, both males and females, and infanticidal males. And what we found is that there's one particular brain area in this area called the medial preoptic area, in which we see enormous activity as seen by the very high expression of CFOS. Now, this is not exactly a surprising result. It has been shown in the 50s already that the medial preoptic area is essential for maternal behavior. So what's new here is that, well, it's not only maternal behavior, it's also essential for paternal behavior. And the question is, what are these neurons? There are many things happening in the medial preoptic area. It's an area that controls thermosensation, mating behavior, all sort of other behavior than parental behavior. So what are these neurons? And in study that I won't detail, we were able to identify a particular marker, the neuropeptide galanin, as expressed strongly in these activated neurons during parental behavior. And because we identify a marker, we can now do manipulation, genetic manipulation of the behavior. And the idea is that what if we kill these neurons? Are parental male and female still parental? Or what if we take a non-parental animal, an infanticidal male, and what if we activate these galanin neurons? Will this male still be infanticidal, or will it be able to display parental behavior? And so we've done both sets of experiments. In one set of experiments, we genetically kill these galanin neurons in the medial preoptic area, and then we test parental male and female, and what we found is that the ablation of these neurons entirely abolish maternal and paternal behavior. And in virgin female, it also elicits infanticidal behavior. So that suggests that these neurons are essential for the display of both maternal and paternal behavior. So now we take an infanticidal male and we optogenetically activate these neurons in an infanticidal male, and then look at the behavior of this male with pups. And what we found is that males that are normally infanticidal now become paternal when we activate these galanin neurons. So in just one slide, I summarize a lot of work that I think convincingly demonstrate that these MPOA galanin neurons are both necessary and sufficient for the control of parental behavior, and this is true both in the male as in the female brain. So if we go back to our model, I think that we clearly demonstrate that both the male and the female brain have circuits that underlie both male and female behavior, and that these enable the animal to have enormous behavioral flexibility, which is occasionally a female will behave like a male, and occasionally a male will take behavior that are normally displayed by a female. And this actually had been already described many, many years ago in the 20s and 30s. And this was before the discovery of steroid hormones. And this very famous behaviorist noticed that in any species they looked at, there were males that occasionally behave like female and female that occasionally behave like males. And at the time, nobody was talking about neuronal circuit, but they said there must exist the innate organization essential to the mediation of the mating pattern of either sex. And it's actually interesting that with the discovery of sex hormones, suddenly males behave only like males and females only behave like females. So suddenly the categorical discrimination between males and females became absolute, when before that was not that clear. Now, moreover, if you look at other species, for example, fish, reptiles, or even flies, it has already been shown that in some circumstances, male can switch to behavior of female and female like a male. I won't have time to describe this, but these are very established studies. But just the assumption was that these had disappeared during evolution is no longer true in mammals. And I don't think that's the case. And indeed, as I mentioned, very recent studies in human brain suggest that there are no categorical differences in male versus females, and that instead the human brain is a mosaic with sex-specific features. So just to conclude, I would like to cite some of the work of two psychologists, which I think show some very strong similarity with the model I presented here coming from the mouse. I don't know if you can see, or is it a bit dark? So Melissa Hines, a psychologist from the University of Cambridge, I think convincingly demonstrated that the behavior, male and female behavior in human, is more of a continuum than something just that is categorical in males or females. And this is true both for morphological traits such as heights. So according to the heights of somebody, you can't really determine for sure whether the person is a man or woman. Obviously, at lower height, it's more likely to be a woman. Higher height, it's more likely to be a man. But there is this enormous overlap in between. Similarly, there's exactly the same type of curve if you look at children playing with boy toys or girl toys. And then these differences become a little bit more pronounced, actually quite more pronounced in the adult, but it still show a significant overlap. Now, the idea of a continuum is quite interesting, but where is the role of hormones here? And again, Melissa Hines, looking at congenital adrenal dysplasia in which young girls are exposed to very high level of testosterone, notice the following thing, which is that if you look at the percentage of women that wish to live as men, it's very low in the normal population, and it's significantly higher in women that have been exposed to prenatal testosterone. So sex steroids clearly have a role in gender identity. But I would like to point your attention to the numbers, which is that we go from 0.01% to 2%. So that still does not explain 98% of these patients who have been exposed to testosterone and still wish to live as women. So steroids have a role, but those roles might be quite different on different type of behavior, and as far as gender identity, it seems that they play a role, but that is not the only issue. Now, last slide, I would like to show some of the data of Janet Hyde from the University of Wisconsin-Madison that look at gender differences in cognitive abilities. And we all hear that men are better at math, but actually if you look at large data set, those differences literally disappear. So the only way somebody is able to find differences is to look at a small population at one particular age in one particular condition. But if you expand this to the general population, these differences disappear. Similar for verbal performance or spatial performance, large data sets show that there is very little difference. I think what is really important also is to see how the assumptions are evolving. This, I think, is a very interesting data plot that shows the percentage of math PhD awarded to women in the US. As you can see, in the 20s and 30s, they were up to 15% of women of PhD in math given to women. And then in the 50s, women were staying at home and taking care of the household, and now the percentage goes here, and now is going up again. And so I think there's quite a lot that has to do with our environment and with the expectation, and I would like simply to finish with a story that actually happened here at MIT, and I found very interesting. Many of you know Barbara Barris, who is now the chair of neuroscience at Stanford, and when Barbara gave a talk many years ago here, she, a very prominent scientist from MIT, turned to his neighbor and say, he is such a much better scientist than his sister, and he was obviously commenting on Barbara Barres, who was Ben Barres 20 years ago, after he decided to live as a man. And so these, I think, very beautifully illustrate assumption and stereotype on evaluating people of different gender. So to finish, I think some of the emerging idea that I presented is that the very simple dichotomy between male and female brains or male and female behavior is largely wrong. There are a lot of shared circuit, and it's more of a continuum than a dichotomy. I think that this being said, gender identity is something very strong that emerged very early in humans. Infants that are 2 or 3 years old are able to express the fact that they know they are boy or they know they are girl, even if sometimes the biological sex does not fit. So that's something we do not completely understand, or we do not understand at all, but it clearly is only partially steroid dependent. And as I mentioned, I think one of the key will be to be able to really have better inference on the function of the brain from a structure, neural structure, and then finally to get rid of quite a number of gender stereotypes. And here are the people who did the work and had an enormous influence in all the research in my lab. Thank you very much.